Oh, oh, there we go. Hi. I think everything is working. Wait, I'll keep myself like this. Oh, golly, I forgot I raised up my camera. It's okay, we're gonna get started. Oh, that's so bright. We made it work. All right. So I need to figure out how I want to actually start these. This is episode 1. This is... My camera's about to fall over. Oh, good golly, Miss Molly. Okay. Oh, that's why everything looked weird. Haha. Okay. Awkward start because I don't know how to start this. Okay. I think I need to have like a thing that actually starts it. God, my lights are so frustrating. All right, we are live, so let's start. Fuck it, let's just start right now. So, hey there, biologists. Today is the first live episode of WildEcoEvo. I'm calling it the Learn Adventurously podcast. I'm not sure what we're going to keep calling it, but WildEcoEvo is going to take you through some of the most interesting conservation, biology, ecology, evolution, and other nature-related stories and articles and research from the week back, the week ahead. We don't know the future yet. Anyways, today's inaugural episode, is going to focus on 3 main topics. 1, how we can actually use ants in Gabon, to detect viruses. We're going to talk about the rising deforestation inside of the Brazilian Cerrado, and we are going to talk about fluorescent frogs. Fluorescent frogs is what we're going to talk about. And oh my god, anyways, let's get started. So, let's get started with our first story. So, I found this earlier today. It's called, Do Virus Detecting Ants Hold the Key to Preventing Zoonotic Diseases? So, this really spiked my interest because virus detecting ants, very interesting. So a zoonotic disease is a disease that is passed from from animals to humans. It is passed along. These are really well-known ones, such as Ebola, such as, COVID was one, or potentially one if it was spread through bats. Rabies is probably the most well-known example. So, if we break down what this article is actually getting into, it really discusses a study that found that certain species of ants may help us to find and detect zoonotic diseases. But how can they do that? So this story takes us to Gabon, out in Africa. And it is a very interesting way of using ants to detect zoonotic diseases. And what we're looking at are these army ants. So these are in northeastern Gabon. This is from I believe the article authors actually gave these photos. And army ants are well known for having, going through an area with a big army and then just sweeping through and pretty much eating everything that lives. So if there is insects in their path, they will eat them. If there is a dead mammal, something that had died and it's sitting there, they're going to eat it. There are also cases, I'm not sure with this exact species, but I believe it was an army ant that it does happen that they do eat on occasion humans, especially small babies that can't run away because they can't run yet. I shouldn't smile about that. That's not funny. But they'll actually suffocate them to death. They'll literally crawl into them. Very morbid start to this. Anyways, But think about that. They eat absolutely everything in their path. Mammals, insects, arthropods, reptiles, large animals, they will eat them. So what the researchers did was collect these ants and analyze their stomach contents for evidence of viruses. Because think about it, if an animal dies and it has rabies, the ants eat that animal, the ants will then have that rabies virus inside of them. Even if it doesn't affect them directly, they will still have it present inside of their body. They could just be silent carriers. They could be essentially viral banks. So, with this idea in mind, they looked at 200 army ants from 29 different colonies, and they found 50,000 viral sequences belonging to 157 genera and 56 families. An insane number, but also diversity of viral sequences. And you may be thinking, well, yeah, of course. Anything viral, bacterial, anything tiny, there's usually a lot of diversity. There's usually plenty that we do not expect. But the authors decided to take it one step further, and they looked and tried to classify all of these viruses. They found around half of them were previously undescribed to science. Half of the 50,000 viral sequences found were not previously known to scientists. And this is really, really important for identifying new zoonotic diseases because, especially in this area, between 2012 and 2022, zoonotic epidemics such as Ebola, monkeypox, Marburg viruses rose by 63% compared to the previous decade. So by actually looking at ants, which are, people don't really care about them, right? If we're having to go out and say we're sampling every rodent in an area to look for zoonotic diseases, rodents are often carriers of zoonotic diseases or carriers of carriers, like in the case of the bubonic plague, Yersinia pestis. It was the fleas on the rats that were spreading the disease, not the actual rats themselves. So, why do ants instead? Well, rats are kind of hard to find. They can be kind of tricky to find. Also, people do tend to get a little squeamish whenever we are talking about culling vertebrate mammals in general. There's a lot more approvals that need to be done. And we're also not getting a wide suite of data. Ants, these guys, like we said, they're eating everything. And they're omnivores, so they're also eating plant material, but they are eating individuals that would be carriers of zoonotic diseases. They're actually eating the rats. They're eating everything else. And no one cares if you kill ants. No one cares if you go into an ant colony, scoop up 300 of them and say, I'm going to grind them up and look for viral sequences. No one cares. So it's much easier, and arguably, because there are, in some ants, there's colonies 1 million strong, taking 300 is not a big deal. Point blank, it's just not a big deal. So, in essence, by using ants, we can actually detect viral sequences. Now, the authors do hope that they are able to actually use this information, not just say, hey, we found viruses. They actually want to use this information in order to combat zoonotic diseases, to identify these brand new viruses that could affect us, all through ants, all by using ants. And so I just thought this was a really fascinating story, especially in light of the COVID pandemic, of Ebola, of other large zoonotic diseases that do come into play. Now, this is a multi-part series. WildEcoEvo has more than one story. You see how I'm drumming this up for dramatic effect because I'm not really sure how to transition from one story to the next. So let's do that. I want to talk a little bit. Let's have a big transition really quick. Because I want to talk about the Cerrado. So we all know about the Brazilian Amazon. Giant, incredible rainforest, captures an insane amount of the world's biodiversity, critical to our planet's climate, critical to many ecosystems, critical to many indigenous peoples in those areas. But people know about the Amazon rainforest, but they don't really know about the Cerrado. And I find that again and again. Brazilians know about the Cerrado for sure. They always know about the Cerrado. What the Cerrado is, let me see, I think I actually have a, yes. So this is going to be an important thing. The Cerrado is actually the world's most biodiverse savanna ecosystem. Let me say that again. The Cerrado is one of the world's most biodiverse places and probably one of the most biodiverse grassland savanna ecosystems we have on this planet. Think about that. And it's not a small area. It is massive. It is beautiful. There we go. This is what I was looking for. 200 species of mammals, 800 species of birds, 180 species of reptiles, 1,200 species of fish, 11,000 plant species found, where half of those are found nowhere else on this planet. Think about that. There are thousands of species in the Cerrado found nowhere else. And it's also important for people. I think so often in conservation, we forget about this human aspect that we're trying to conserve. We focus on the animals that are going extinct, the plant life that is getting decimated, but we still, to this day, turn a blind eye to the people. The Cerrado is an important source of water and electricity. Okay? Millions of people depend on water that stems or is through the Cerrado, and that water is used in hydroelectric facilities to actually produce electricity for people. 9 out of 10 Brazilians utilize water generated from water originating in the Cerrado savanna. I messed that up. Whatever. We're going with it. Now, what's happening in the Cerrado? Well, much like its Brazilian counterpart, the Amazonian counterpart, it is being deforested at massive rates. In fact, I believe it's this page. Yes, here is a visualization from 1984 up until 2022 showing deforestation in the Cerrado. This is one small region, but you can see that what started out as green, in only 30 years, there's almost none left. And I understand deforestation may be habitat degradation because in reality a lot of this is grassland, so there's no forest, I guess you could say. I guess it's a forest to ants, but not to us. But what is actually going on here? Why is it actually surging in the Cerrado? Because deforestation is becoming a greater and greater effect. And in the Amazon, deforestation is actually declining. In the first half of the year, we've actually seen deforestation rates plummet, partially due to new political influences and also the fact that people have been screaming to please stop destroying the Amazon for decades. But at the same time, ignoring the fact that the Cerrado is losing around 8,100 acres per day. Per day. This year, the Cerrado has lost 8,100 acres a day. In hectares, that's about 3,200 hectares. And the leading cause? Monocrop agriculture. Monocrop agriculture like soy production. And let's be real, soy production, not really for tofu. People love to say that. It's like vegans are destroying the planet because of soy production. No, that soy is going to feed cows. It's an insane amount of water being utilized to go feed cows, which then themselves need an insane amount of water. So, weak arguments aside, let's just push those down at the start. But it's a very tricky part because this incredible area, again, is being deforested. And we can see that it is lower than it used to be. 2000s, I mean, 90s through the 2000s were a crazy bad time for deforestation in general. But the Cerrado here, you can actually see the size of the Brazilian Cerrado. It's taking up a decent amount of the country. I mean, this is not a small ecosystem, and people do overlook it like crazy. But you can see that it's actually rising. And what these authors are stating is that because there is now less deforestation in the Amazon, developers are still needing to, well, develop, not needing to, developers want to develop. And they're now developing in the Cerrado, where some, if I remember correctly, I have it in my notes somewhere, but there is, because there are economic incentives to developing Cerrado, the Brazilian government is dissuading Amazonian deforestation and at the same time supporting Cerrado deforestation. But yeah, I just really wanted to bring this up because yeah, I just think the Cerrado is an important area that gets really overlooked. And when we talk about the Amazon deforestation, I mean, this is sort of a thing in general, right? Where people like to ignore grasslands, they are often overlooked. Yeah. And now I'm just spinning my wheels, trying to talk about this more. But I think it's time for our next story. And this is the first one. I don't care if it's bad. I do want to actually get a co-host on this. I do want to get a second person that we can actually have on here and bounce ideas off of. But I realized I've had this idea now for this show. Maybe a little aside, maybe building tension, maybe I'm stalling for time, who knows. When I started this up, I wanted a second co-host, but I realized I was waiting for that second co-host forever. Just waiting and waiting and waiting and waiting and waiting, and then not actually making any content. So this is me squashing that perfectionism and doing it anyways. And I don't know why I'm telling you this. I don't even know if you're there. But one day this podcast will be fire. Currently, it's on fire. So, let's go to the one that I am most excited to talk about. Biofluorescent frogs. Seriously. So this is something this is a concept I've been following for a little bit now. Back in 2017, there was, this paper from Taboada et al. Titled Naturally Occurring Fluorescence in Frogs. And this was very, very fascinating to me. The frogs, here we go, here's some great showcases. So what fluorescence is? Fluorescence is very similar to a glow stick, okay? There is something that is causing these frogs to be brighter than their surrounding environment. So if a frog is sitting next to a rock, for example, and it's twilight, the frog would be a little bit brighter than the rock, and that's how we'd be able to detect fluorescence in one example. It may not be much brighter. They're not like a light bulb sitting next to it. Think of it like a very weak glow stick. I almost said glowfish. That's not right. Think of it like a very weak glow stick. And the current thought, this is still all very new information to us, but the current thought is that this fluorescence actually helps frogs find each other during twilight conditions when they are at their most active. Again, frogs are most active during twilight. And so if they're able to fluoresce just a little bit, even if it's a minuscule amount, and the other frogs are able to detect them, then that's how they can find each other at nighttime. So they don't necessarily need perfect night vision. They don't necessarily need their calls to find each other. And there are plenty of frogs that really don't call that often. and there's plenty that just kind of wander around. And we're still trying to understand exactly why calls are calls. But this is something I have been following for quite a while because I believe that frogs' visual systems, as well as their communicative systems, are vastly, vastly, vastly overlooked. We've detected in the past UV reflectance on frogs, just like we have in birds, right? We know that birds can see in that UV spectrum, and I have a very strong feeling, hypothesis, theory thing that makes me think that frogs do also see in UV, and there's a few studies that back it up, but not enough to be conclusive. So, this is the old paper. This is the 2017 paper that said, hey, we found fluorescence in this frog. What did we do? We looked through them. We tried to figure out why they're fluorescent. And what they found was several chemical compounds. So think of it like a glow stick, actually. This glow stick analogy is perfect. Look at this. This is that fluorescing frog that is actually just kind of jacked. But think about it like a glow stick, okay? So say we have some glow stick here. And what's inside of the glow stick? Chemicals. Wow, look at that. It is emitting light. Well, the glow stick just doesn't emit light forever. We know that over time, it gets a little bit more dull. We know that if we get a brand new glow stick, it may not be actually good. So, what causes the glow stick to glow? Well, light hits it. Some type of light hits this glow stick, and then those chemicals inside of it will actually capture the photons from this light. They'll capture it and store it, and then over time emit a different wavelength of light. Usually it's a different wavelength. So maybe these glow sticks are absorbing blue wavelengths of light, but through various shifts in wavelengths, they're now emitting green wavelengths of light. And these chemicals are found in their lymph nodes, they're found in their skin, but this has been already detected. So then what is this new article doing? We said there was a brand new article looking at fluorescence. They did a concept looking for ecological tuning and trying to see if these biofluorescent signals are actually adaptive or not. But how do you do that? How do you detect if something is adaptive? There's this group ideology in biology, more so in, no, it's in a lot of people. It's in biologists, it's in the general public, where we think that if a species has a trait, it must be adaptive for it. This is called adaptationist thinking. But it is fundamentally flawed. But why is it? Well, why is the assumption that an adaptation must be good for the organism? Right? I mean, we've seen organisms before. Think about the cows that get born all the time that have extra appendages. Well, if you found it in the wild, we wouldn't assume that is a positive adaptation. We wouldn't assume that's a good trait for natural selection. But we do that with so many organisms. The giraffe's neck must be there so that it can reach the tops of trees. The frogs fluoresce because it must be good for them. But there's this concept called evolutionary spandrels. Sometimes traits are just there because they're just there. Sometimes traits are exhibited on an organism because of something that happened maybe way in the past. Or maybe it's just there for no real reason and it's completely neutral. Maybe it's even a little bit bad, but past effects of their evolutionary history made those traits stay. I talked about this with Boletoglossus salamanders. They have these incredible webbed hands. And for years, for decades, it was said that, oh, these webbed feet enable them to suction cup climb up leaves in the trees in order to exploit a new niche. That's how they were able to colonize throughout Central and South America. But a recent study looked into it, and they found that this isn't adaptive. This isn't adaptive whatsoever. In fact, the only reason those salamanders have those webbed feet is because it's a little bit warmer down south. So they develop faster. So their feet are unable to actually split, just like we in the wombs have webbed fingers and webbed toes. If we get born very premature, we may still have that webbing. And in certain cases, there's genetic defects that also cause that webbing, but that's besides the point. So in those cases, with these salamanders' feet, it was not actually adaptive. It was just sort of a leftover. It just sort of was there because of their evolutionary history, because of going down further south. So then, how can we test this fluorescence inside of frogs? Is it adaptive? Is it actually adaptive? And that's what this paper is looking at doing. Now, there's no pretty pictures inside of this paper. So this is also a preprint, meaning that it has not been peer-reviewed yet, but it is open for everyone to view and make their own conclusions while it is still getting that peer review. I will say that things do change during peer review, so keep it with a grain of salt, but their conclusions I doubt are going to change all too often. So I'm going to keep this up while we talk a little bit more. Now, what the authors did in that study to test for adaptation in this crazy fluorescence in frogs is they had 4 criteria to detect. And you know what? I'm gonna do something neat. Look at this. Copy. Nope. Copy. Image. We're gonna go here. Boom. We're gonna put it... What is going on here? We are going to put this here. We're going to make it big. And then we're gonna write notes. Look at that. Wow. So, let's put the pen down. Cool. So, we have 4 criteria to test for adaptation here. Okay. So, adaptive. Adaptive. 4 criteria. The first. Fluorescent pigment will absorb the dominant wavelengths of light found in the environment. I'm going to say that again. The fluorescent pigment found within the frog will absorb the dominant wavelengths of light found in the environment. Look at that. That was awful. And the reason for why this would be considered adaptive if it is found. Let's write this out so we can all be on the same page, and I'm covering up most of it. Here we go. We'll put myself down here with the frogs. So, why would this be adaptive? Why would it be adaptive? So, let's say that we detected fluorescence in frogs. Okay, they fluoresce underneath a purple light. Well, maybe they never actually get that purple light in their environment. Maybe it's not a big component. Maybe it's a very small portion of the dominant visible wavelengths of light found in their environment. And if that's the case, then this really wouldn't be adaptive. Rather, it would just kind of be this little quirk, this random thing that they can do. Oh, they can fluoresce, but it has no real meaning in nature. Okay, so this is trying to say, do the wavelengths that they fluoresce at actually match their environment? Okay. So then the second criteria. The fluorescence will be viewed by a receiver against a contrasting background. Visible on background. Now, why is this? Well, let's say again that these frogs can fluoresce. We don't need a question mark there. Let's say they can fluoresce. Well, they do fluoresce. So let's say they're fluorescing. Let's say they fluoresce a bright green, but maybe everything in the environment actually fluoresces a little bit of a bright green, or their fluorescence isn't very visible from the background. Then this trait would not be adaptive. Either the frogs would be completely hidden. I mean, look at this. So let's say it's that first scenario, right? And there's a frog. We're gonna denote it by this little circle, and it's fluorescing green. Okay, awesome. But what if everything in the environment is fluorescing green, right? Oh my gosh, I'm hitting buttons. I am so... Can we not do this? Can we not do this? Can we not do this? There we go. Okay, so if everything is fluorescing green, then nothing is fluorescing green, right? That's essentially what this is saying, is can we actually detect it? So maybe the whole background, maybe everything is green and it's very hard to fluoresce, but these frogs, hey, they are fluorescing purple, they are visible, sort of. That's really what this second point is trying to get at. Okay, the third criteria. Let's make it green so we're consistent. Organisms viewing the fluorescence will have spectral sensitivity in the fluorescent emission range. That is crazy jargon, but basically it's saying, can the organisms see it? So this is really saying that inside of the eyes, so inside of eyes in general, they can sense light and color. Look at this. We'll give them purple eyes for no real reason. So they can sense it, but it's because in the back there are rods and cones which enable organisms to see different wavelengths of light. So what this criteria is really getting at is, hey, cool, you have a frog. You have a frog over here. It's fluorescing, and it's fluorescing green. Incredible. But then the other frog that wants to see it literally can't see the color green. So why would this matter? That's what they're trying to test. They're trying to say, cool, the frogs can fluoresce, but are they actually able to even detect it in their biology? Because maybe they're, colorblind, right? Animals do not have the same visual systems that we have in many cases. It's like your dog, right? They can't see, if you throw I think it's a blue tennis ball out into a green field, they can't tell green from blue. I think that example might be wrong, but the concept still applies. So, the final criteria to test if this fluorescence is adaptive or not is that the fluorescent must be on a part of the body displayed during signaling. So, okay, I'm not going to write this out. So, on the body, visibly. Because, yeah, it may be great that the frogs can fluoresce, right? But what if they only fluoresce, like, on the bottom of their feet, and they don't ever raise up their feet and wave to the other frogs and, say I'm here, kind of like if you're at a concert with a glow-in-the-dark wristband. So, these are the 4 criteria that the authors decided to look at to see, hey, we know frogs fluoresce, but is this adaptive? Is this good for their biology? What they found. Let's make this purple. I like this purple. The first one. They absorb the dominant wavelengths of light found in the environment. Was that criteria found? It was. What they found is that the frogs are absorbing majority blue wavelengths of light, or the fluorescence is able to absorb majority blue wavelengths of light, which is the dominant wavelength present during twilight hours when the frogs are most active. So it's a little confusing. They're absorbing blue light, but then are emitting green. That's kind of how fluorescence works. It doesn't emit back usually the same wavelengths. It usually gets changed a little bit, shortened, lengthened, whatever. And then so it's a little bit of a different color, but that's what happens. But for this purpose, the frogs do absorb the dominant wavelength of light found during their dominant activity period. So the second one, visible on the background. They found that this was true. They found that if you looked at the frogs, if you tried to fluoresce them, that they were very, very visible from the background. The third one, can the organisms actually even detect this fluorescence? Now, I've talked about frog visual systems a little bit already. I think they're under-researched. I think there's many issues with them and with the studies, because we often it's still a bias that we look at them with only the human visual spectrum. We ignore their UV, we ignore, more and more we're starting to realize that frogs might be able to see in infrared a little bit, but they found that the frogs are able to detect these wavelengths. And the last one, is it on the body visibly? As we can kind of see on this picture here, it is literally the whole damn body. So yeah, it's visible. So what they found is that this is actually adaptive. They found that it is adaptive. And in fact, they found that fluorescence contributes 18 to 29% of the total emerging light under twilight and nocturnal scenarios. Basically meaning that these frogs are around 20%. No, that's not even the right way of saying that. I was about to say they're about 20% brighter, but that's not the case. It's 20% of the light that is bouncing off of the frogs is because of this fluorescence of glowing in the dark. Now, the title of this article mentions ecological tuning. And that was a phrase that I have seen a couple times, but didn't really know. So I decided to look it up. Decided to understand it more. And it does fit very well with this system. So ecological tuning. Let's, okay, I'm not using the pen. I would like to move things. There we go. Let's keep the frogs on the screen because we are visual creatures by nature. Ecological tuning. This intimately relates to the 4 criteria listed above. Okay. Ecological tuning is essentially where there is some adaptation that is being adapted to local environments. Okay, think about that. You have some adaptation, and then in different environments, it is tuned slightly differently in order to account for it. That's also a great evidence of this adaptation. That's actually what these 4 criteria are looking at. Is this ecologically tuned? And think about this first one. Absorb the dominant wavelength. Let's say these frogs were active in a different part of the world, a different time, and maybe the dominant wavelength during that time is red. I don't think that's actually biologically possible, but let's say it's more red than where they're at now. We would expect that the fluorescence is also a little bit more red in this different environment because the adaptation is tuned towards local environments. And they were able to detect that. So this study was really fascinating. I really enjoyed it. Again, it's still preprint, so there's really not much in terms of the way of great pictures. This is all very visual spectrum stuff. No, I don't even know if there's any frog pictures. Something interesting too is they also found that a lot of individuals had an orange peak in their adaptation. So they had an orange wavelength that was being emitted on the fluorescence. And this is actually maybe a great example of a spandrel. Maybe there was some evolutionary force 3 million years ago where frogs that had an orange wavelength were more able to survive and pass on their genes. So therefore, they did. But yeah. Oh, here we go. Here's some frog pictures. So yeah, we can see that the fluorescence. Let's get rid of that. Oh, I should have had this picture up the whole time. Can we not? I want to see the picture, not the caption. God, they have so much money. Actually, I don't know how much bioarchive has. Okay, can we? Can we not? There we go. So yeah, we can see all of the fluorescence here. We can actually see, like, I think one that's really cool is this Boana lanciformis in the center. Look at this pattern that it has for its fluorescence. Let me see. Can we zoom in? Yeah, we can. I am so annoyed by this. Wow. I just want to open image in new tab. That's what we're going to do. There we go. Of course, it's terrible. Wow. Hold on. This one, open image in new tab. There we go. So yeah, we can see that Boana lanciformis right here. It has these really cool patches underneath. And I need to look into more, but I mean, you can all see the fluorescence on the lips. You can see it on the sides. These are absolutely visible. And then we can see it also in other ones like Synax, Strigulatus, Hamptophrine boliviana, Boana atlantica. There are some very fascinating realms to explore. Or if you look at Boana calcerata, its eyes are the one fluorescing. Incredible, right? So there's incredible variation in frog fluorescence in terms of where it is occurring, its pattern, the way it looks, how it fluoresces, and I'm sure there's so much that we don't even know yet. We don't even know enough to test it properly yet. But this is something I really wanted to show all of y'all today. I thought this was an incredible paper, study, finding, and yeah, just want to show it. As always, links will be down in the description once I post this live. But yeah, I'm going to, yeah. So that is actually everything I had planned out for today's inaugural episode. In the future, I would love to get a co-host. If there's anyone you want to see on here, let me know. I'll try to get them on. Hopefully we can. And I mean co-hosts in the sense of changing. Like, meh, we'll just get a random bench. But anyways, that's everything for today. So thank you. We'll be trying to do these once a week. So tune in maybe next Friday for another series of really cool studies, conservation news, and other stuff. Yeah. So